In today's class, let us try to learn about very important concept, that is the enzymes which are used in the hospitals and the importance of enzymes, biological agents that are essential for treatment. In the enzymes, the very important enzyme is hyaluronidase. We have chemotrypsin, alpha chemotrypsin, Seratopeptidase, Eurokinase, tissue plasminogen activator, and Asparaginase. These enzymes are very commonly used in the hospitals, and they have certain clinical significances, clinical uses, and some enzymes are claimed to be having different pharmacological actions. Let us try to understand about these enzymes, their uses, and certain clinical points in the hospital. So we will start with hyaluronidase enzyme. Hyaluronidase enzyme is very important enzyme in the hospital sector, because this enzyme is important in metabolism of hyaluronic acid. So hyaluronic acid hyaluronic acid is important component of different tissues. It is the important component of extracellular matrix of certain tissues. It is important component of extracellular matrix of connective tissue. It is important component of joints. It is very important in joints. And hyaluronic acid is also important component in skin extracellular matrix. Hyaluronic acid is very important. So hyaluronidase is enzyme. This enzyme, hyaluronidase enzyme, tries to break down this hyaluronic acid. When hyaluronic acid is broken down, the extracellular components, tissues will be degraded, and in that, tissue permeation increases. It causes tissue permeation or tissue permeability. And with that, on the tissues, if certain drugs are used, on skin, if certain drugs are used, it increases the penetrating power of different drugs, because the hyaluronic acid was broken down and the uh, tissue viscosity decreases, and with that, the drugs will easily transfer from one place to the other place. And important point, hyaluronidase enzyme is obtained from the mammalian testis. From the mammalian testis is obtained, and it is available as topical cream. It is available as injections, which is used as subcutaneous, and it is also used as injection in intramuscular route or intrathecal, intraarticular. In joints also it is used. If joints are stiffed due to increased hyaluronic acid, certain joint extracellular matrix will be increased, and in that cases we use intraarticular hyaluronidase enzyme, and with that, the hyaluronic acid extracellular matrix will be broken, and it decreases the viscosity. Okay, so this enzyme is important. And other important points, let me add here. Yes, as I was explaining you, the actions of hyaluronidase enzyme depolymerizes the hyaluronic acid, because hyaluronic acid is the major component of certain connective tissue. Okay, in connective tissue, this hyaluronic acid increases the cell density. For example, see, if we imagine the skin, this is the skin cells. In the skin cells, the extracellular matrix was the main component of hyaluronic acid. Okay, when hyaluronidase enzyme is injected here, the extracellular matrix of hyaluronic acid will be broken down, or it will be digested, or it will be depolymerizes. Depolymerization of this hyaluronic acid will be caused, and with that, it will decrease the cell matrix density. When cell matrix density decreases, it causes tissue permeability. Tissue permeability. It increases tissue permeability. So this is the important mechanism of hyaluronidase enzyme. Now let us see the important uses of hyaluronidase. It is very important in promotion of certain drugs absorption. Some drug absorption is promoted when hyaluronidase enzyme is given through subcutaneous route, intramuscular route, or intraarticular route. It is also important in resorption of extravasated fluid. When certain fluid is accumulated in certain tissue, and this fluid is unable to transfer into the blood vessels, okay, or in the lymphatic vessels. So in that cases, if the hyaluronic acid matrix is very dense here, it is very difficult for the accumulated fluid to transfer into the blood or into the lymphatic vessels. So in that case, if patient is having edema, or if patient is having hematoma, in that cases, if hyaluronidase injection is injected here, this tissue density decreases and the accumulated hematoma or extravasated fluid will enter into the blood and it decreases edema. So in edematous condition also, hyaluronidase enzyme is useful. And the third very important use of hyaluronidase enzyme is it facilitates the diffusion of local anesthetic agent. In the same, see, when anesthetic agent is injected here, if the extracellular matrix is very dense, in that cases the anesthetic agent can't diffuse into the other tissues. So in that case, if this local anesthetic is added with hyaluronidase enzyme, the cell matrix density decreases, and with that, the anesthetic agent will easily diffuse into the other areas. So simple point, remember, hyaluronidase enzyme decreases the cell matrix density, and through that it improves the tissue permeability, and with that it decreases tissue density. And in that cases, if certain drugs are used, they will be easily they can easily transfer from one area to the other area. So this is the use of hyaluronidase enzyme. And now let us discuss about chemotrypsin, very important enzyme which is used in the hospital. Chemotrypsin is mainly obtained from ox pancreas because it is obtained from pancreas. See, trypsin, chymotrypsin, alpha chymotrypsin, all these are proteolytic enzymes. Proteolytic enzymes, and these enzymes are major constituents of pancreas. So in pancreas, all these chymotrypsin enzymes are available as chymotrypsinogen. They are available as chymotrypsinogen. When patient takes food, let me explain you. When a person takes food, when person takes food from the stomach, when food enters into the small intestine through duodenum, here when food enters into the duodenum, one very important element is released from the body, and that is CCK. When patient takes food, when food enters into the duodenum, CCK is released from the body, which is CCK is called cholecystokinin. CCK is called cholecystokinin. This cholecystokinin releases when food enters into the duodenum, and this cholecystokinin will go to pancreas, and in pancreas it will stimulate the release of chymotrypsinogen. Chymotrypsinogen. Trypsinogen. This chymotrypsinogen from the pancreas, upon activation of cholecystokinin, this will release into the duodenum. And in the duodenum, this chymotrypsinogen, it will be activated into chymotrypsin. Chymotrypsin. And this chymotrypsin is activated in the presence of trypsin. This trypsin will convert the chymotrypsinogen into chymotrypsin, and this chymotrypsin now it is available in the activated form in the duodenum and in the small intestines. In the small intestine, this chymotrypsin will break down the proteins. So it breaks down the proteins. Proteins are converted into amino acids, and these amino acids are absorbed from the small intestines into the hepatic portal vein, and it will be taken to liver here through hepatic portal vein from the small intestine. This is the function of chymotrypsin. Now, in certain conditions where patient is having pancreatitis or pancreas dysfunction or failure of the pancreas, in that cases, chymotrypsin is very commonly used in the hospital. Now let's come back. It is available as oral tablet because chymotrypsin is important in digestion of proteins, and it is also sometimes available as topical ointment on the skin. On the skin, certain proteins are metabolized because in certain healing conditions and in psoriatic conditions, few conditions, the skin will have a lot of proteins which are to be removed from the skin or from the tissues. So in that cases, if topical chymotrypsin is used, they will degrade the excess proteins. So, however, this topical chymotrypsin is very rarely used. Mainly, we use chymotrypsin as tablets for pancreatic disorders, and these chymotrypsin tablets are available along with the trypsin, okay, in combination. Anyways, this trypsin converts the chymotrypsinogen into trypsin that we have already discussed. Now let's discuss about the next very important enzyme, which is called alpha chymotrypsin, which is very similar to chymotrypsin. Here, one difference is alpha chymotrypsin is not only available in tablet form, but also it is available in IV form. This is very important. And this chymotrypsin, alpha chymotrypsin, has certain uses in the hospitals. This alpha chymotrypsin is used as certain conditions. It is used as mucolytic agent, because it is having mucolytic action. It is used as anti-inflammatory agent, anti-inflammatory agent, and it is used in edema. in edema because it is having anti-edematous actions. And the fourth action, anyways, we already know that it is a proteolytic enzyme. It breaks down the proteins. Okay, proteins. So it is used as proteolytic agents. So these are different actions of alpha chymotrypsin. It is used as mucolytic agent. It is used as anti-inflammatory agent. It is used in edema, and also it is used in proteolytic different conditions where proteolytic action is required. So this is about alpha chymotrypsinogen. Now, if we see the specific uses of alpha chymotrypsinogen, alpha chymotrypsin, it is used in cataract surgeries. During cataract surgeries, it facilitates the removal of lens, and it is used to reduce inflammatory edema due to trauma, infection, and different surgical conditions. Now, next very important enzyme is Seratopeptidase. This is very important enzyme which is commonly used in surgical ward, in surgical conditions, in emergency, and in orthopedic department. This Seratopeptidase is very important. Seratopeptidase is also called as Serrapeptidase. Serrapeptidase. It is also called as serralysine. So these are different names of Seratopeptidase. This Seratopeptidase enzyme is produced from the enterobacterium, which is called Serratia marcescens. Okay, Serratia species of different bacteria it is obtained, and this bacteria is enterobacterium, which is available in GI tract. So Seratopeptidase is available as, particularly it is available as tablets, and these drugs are used along with other NSAIDs like aceclofenac, paracetamol, and different diclofenac and all conditions. It is also available as single formulation. If we see the different actions of this Seratopeptidase, it is claimed that it is having proteolytic activity. It is also claimed that it is having anti-inflammatory actions. That's why it is commonly used in inflammation due to surgery, trauma, infection, chronic conditions like osteoarthritis, rheumatoid arthritis, and different other inflammatory conditions. But for this action, anti-inflammatory action, it has no sufficient evidences. So certain clinical trials are again going on this Seratopeptidase enzyme. Since it is not having enough evidences, we can't directly say that Seratopeptidase is used alone for anti-inflammatory actions. That's why this Seratopeptidase is used along with other NSAIDs. Okay, it is very commonly used only in combination with different NSAIDs. Okay, it is having proteolytic activity, it is having anti-inflammatory activity, and these two activities are just claimed. And however, it is widely used, and many people are having satisfactory benefit reactions. It is used in the hospitals. Okay, so this is about Seratopeptidase, and next we'll discuss about Eurokinase. Eurokinase is another enzyme. Eurokinase is very important enzyme which is having different actions. It has it has fibrinolytic action. Okay, Eurokinase is having fibrinolytic action. Actually, it breaks the fibrin threads which is formed in the coagulation cascade when the clot is already formed. See here, just you can see the clot formation. Clot formation is in the coagulation cascade. Fibrin activation and these fibrin threads plays very important role in finally getting non-soluble fibrins and non-soluble clot. In that, these fibrins are very important. And fibrin breakdown occurs due to urokinase. Urokinase is one enzyme which breaks down these fibrins. Now, let us try to understand the common physiology of coagulation cascade. I think we have already studied this. We have intrinsic pathway and extrinsic pathway. These two pathways has different mechanisms. Starting in intrinsic pathway, starting from the 12th coagulation factor, and in the extrinsic factor, starting from the 7th coagulation factor. At the end of these two intrinsic and extrinsic pathways, they form factor 10A, which is the activated form of coagulation factor 10A. This 10A converts prothrombin into thrombin, and this thrombin again converts the fibrinogen, which is inactive protein, into fibrin, which is active fibrin, which helps mainly in stable fibrin clot formation. So here these fibrins are mediating into the formation of clot at the end. So here these fibrins are broken down by uh, fibrinolytics, and urokinase is one example, best example for this fibrinolytic action. Okay, so urokinase is mainly obtained from the fetal kidneys. Okay, in fetal kidney cell culture it is obtained, and it is having fibrinolytic action we already discussed. Okay, and this mainly, if we see the urokinase uses, it is used mainly in two conditions. Number one, deep vein thrombosis. It is one of the coagulation disorder, and deep vein thrombosis sometimes causes another complication which is called pulmonary embolism. So it is also used in pulmonary embolism. So urokinase injections are used in deep vein thrombosis and pulmonary embolism. About this deep vein thrombosis, I have already posted one video. You can see in the description. I'll add the link. So that is about urokinase. I think you understood about urokinase. Now, next very important enzyme is tissue plasminogen activator. This tissue plasminogen activator is also having same action like urokinase. As we have already discussed, urokinase has fibrinolytic action. Fibrinolytic action. And in tissue plasminogen activators, actually, if we see, we have certain examples like reteplase, altiplase. All these are tissue plasminogen activator, which are derived from DNA technology, rDNA technology, recombinant DNA technology. And these drugs are again used for deep vein thrombosis. They are used for pulmonary emboli and different other conditions where patient has hypercoagulability, and in stroke conditions also, in especially in ischemic stroke, or in patient who is having stroke due to coagulation. So you can see the formulations, they are mainly available in IV form. And yeah, I was explaining you, it is having fibrinolytic activity, same like urokinase, and it is used in acute MI, myocardial infarction, deep vein thrombosis, and pulmonary embolism. Okay, and now let's the very important enzyme is L-asparaginase. This enzyme is very important. First of all, let us see this L-asparaginase has two uses. Number one, clinical use. Clinical use. In clinical uses, it is mainly used in two conditions. One is in cancer, that is ALL, which is also called acute lymphoblastic leukemia. Acute lymphoblastic leukemia, and it is also used for LBL, which is called lymphoblastic lymphoma. Lymphoblastic lymphoma. This is the clinical use. First one, clinically, L-asparaginase enzyme, which is available in IV form. It is used for acute lymphoblastic leukemia, and it is used for lymphoblastic lymphoma. And second very important function use of asparaginases, it is used in food manufacturing industry. In food manufacturing industry, it is having great importance. I'll try to explain its use. See, actually, the action of this L-asparaginase is it will stop the conversion of L-aspargine amino acid into acrylamide. When food is cooked, the food is containing L-asparagine. This is the basic amino acid, and we have sugar moiety. When this amino acid and sugar moieties are heated, or when it is cooked, it will form a Schiff base. This Schiff base is again cooked at high temperatures. It will form a dark colored substance. In this dark colored substance, there is one very important compound which is produced is acrylamide. This acrylamide is the product which is obtained especially in food items where food is cooked at high temperatures. When in high temperatures, the food is converted into black colored substance. This black colored substance is the substance in which acrylamide is one form. This acrylamide is considered as carcinogenic agents. So in food industry, if L-asparaginase enzyme is used, this enzyme will stop the conversion of acrylamide. And in this process, actually, the L-asparaginase is converted into aspartic acid and ammonia. Actually, this L-asparaginase enzyme converts aspargine into aspartic acid and ammonia. The simple action, let me tell you the action of L-asparaginase. In this, the main action of this L-asparaginase is it converts the asparagine amino acid into aspartic acid plus ammonia. This is the conversion of this amino acid. But in the absence of this L-asparaginase, what happens? This asparagine amino acid is converted into acrylamide, which is the carcinogenic agent. That's why in food industries we use this enzyme to bypass this conversion. Actually, the conversion of this asparagine into acrylamide is Maillard reaction. Okay, this Maillard reaction is the reaction in which the asparagine amino acid converts this amino acid into acrylamide, which is the carcinogenic agent. So in this, to bypass this Maillard reaction, we use L-asparginase enzyme, and this L-asparginase enzyme converts the aspargine amino acid into aspartate, which is very safe, and ammonia. Okay, so this is one mechanism of L-asparginase enzyme. And in clinical use, if we see in clinical use in certain cancer conditions, as we have discussed, in cancer like ALL, and LBL, in cancer, the neoplastic cells will depend on asparagine amino acid for its survival. Okay, the normal cells will not depend on the external sources of asparagine, because normal cells can synthesize their own amino acid, but the neoplastic cells can't synthesize their own asparagine. In that cases, if L-aspartate enzyme is used in cancer conditions like neoplastic conditions, these neoplastic cells will try to use this amino acid asparagine, and this asparagine availability is decreased when L-asparginase enzyme is used. This L-asparginase enzyme immediately converts the available asparagine into aspartate and ammonia, and the neoplastic cell will have deficiency of this amino acid, and with that the neoplastic cell will die. So in cancer conditions, L-asparginase enzyme is used to kill the neoplastic cells by decreasing the concentration of asparagine amino acid. This is the mechanism of L-asparginase. So that's it for today. I think you understood the different enzymes that are very useful in the hospitals. I think you liked the video. If you learned something from our video, please don't forget to like, share, and subscribe the channel. Thank you for watching.